you're not winging it. You know, in this case, we have done research using similar molecules. We have some basis for projecting. That's the difference.
It's in a way, it's intended explanation.
Excuse me?
Intended explanation to the problem.
Yes, yes. Now, we have to be prepared for the fact that our research may not agree with our hypothesis, and that's not failure. We'll see, not failure. All right, next thing we have to do is we have to agree on a plan. Critically important, because this plan has to generate the heart of a scientific experiment. And the heart of a scientific experiment, the core, is something called a controlled experiment. And we're going to see what that is. If this is not there, then your testing is really in vain. It's really futile. So, a controlled experiment, it's done under controlled conditions. It means that we are trying to keep all the variables aside except for one. So in this case, our one variable is the shot of medication, new medication, that we're going to give to mice or something like that. We want everything else to be the same. Now, that's not always easy. You know, you can ponder this. You can wake up at 3 o'clock in the morning, as some researchers do, and this will drive you crazy because you think you have pushed aside all the extraneous information so that you can get valid results, but that doesn't always happen. So how do you set up a controlled experiment? So what we're going to do in our method, our imaginary pharmaceutical method, is we're going to go out and establish a plan and decide where we're going to start. We're probably going to start with mice. And we're not going to go out in the field and collect mice in a cage. Why wouldn't that be a good idea?
Because you don't know how they grew up or what they've eaten or how old they are or all the variables.
Exactly. And if they're not all the same genetic signature or different ages, they already may have disease. You know, there's too many variables. So we order laboratory mice. And these have been bred to be as genetically pure as possible. From infancy, conception, they try to control as much as possible all the extraneous variables. And again, that's hard to do perfectly, but it's better than driving mice out in the field. Anything that happens as a result of the independent variable is a dependent variable. So let's imagine this. Let's get down to the nuts and bolts. We have, let's say, 100 laboratory mice. We want a controlled experiment. So we're going to separate these into two groups. A group of 50, which is called the control group, and a group of 50, which is called the experimental group. They're going to be kept under exactly the same environmental conditions. They are going to be fed at the same time every day. They're going to be fed the same food, the water sterilized. We're going to run a series of tests on them every day to see how they're doing. We're going to check them to see if there's hair loss. We're going to put them through a maze to see if their eyesight's being affected. We're going to weigh them. They're obviously all tagged, so we can keep track of each individual mouse, because sometimes there's revelation in that. And we are going to then shave off some of the hair on the back of each mouse, and we're going to inject in them a carcinogen. Sounds cruel. It is, in a sense, but no other method have we come up with to advance to help people. So they're going to develop on their backs a tumor. In the control group, they are going to receive a shot daily of sterilized water. In the experimental group, they are going to receive a shot of this new medication. And we're going to do this for 30 days. Okay, see how we're setting up a controlled experiment. Good place to pause because this is critical. Any questions about why we're doing this and control group, experimental group, independent, dependent variable. So if we've done everything perfectly, anything that happens to these mice should be the result of one thing, the shot. And the control group, theoretically, should progressively get worse. And if the shot of the medication does anything, we should see a change, hopefully a positive change. Last year, I did not see, on a consistent basis, an effort to have a controlled experiment. Some did, but it wasn't always clear. It wasn't clear what their independent variable was. It wasn't clear what steps they had taken to isolate that one variable. Now, some of this is going to be in your writing, your presentation. Some of it is going to be oral. You're going to be questioned. You're going to be queried on this. So even though it may not show up in writing on the board or in your paper, you have to be prepared to say, well, here's what we did. And here's why we did it that way. Again, just some statements again that I extracted from Khan Academy and other places to emphasize the importance of these groups and how they fit in with independent and dependent variable. You're constantly asking yourself this question. Am I isolating a variable? If at some point you say, whoa, we forgot, well, then you kind of have to cancel and start again. So there's evaluation going on during this whole process. You're collecting data on all the mice, by number, volumes of data, hair falling out, gaining weight. Are all the mice gaining weight? Or just certain mice? Or all the control group? How does that compare to the experimental group? I know I'm saying all the time with this, but this is important, imperative that you understand this part. And it was not consistently demonstrated last year. Okay, experiments need to be reproducible. Now, we'll talk about conclusions and how that's important, but no experiment is done once. I wouldn't take a medication that had been tested once. I often wonder when I see these advertisements on TV, medications, and they say, if this happened to you, contact so-and-so, and we got a class action lawsuit. And I'm thinking, wow, was this tested? How did they test it? Who did they test it on? How long did they test it? It's a little unnerving at times to wonder, were they in a rush to get money or beat out their competition, or did they really take the time to make sure that this was producing minimal side effects? Because there are always side effects.